Hello girls, welcome to the online lecture series. So in this online video lecture, let us discuss about influenza virus. So this influenza virus, we all are familiar with influenza virus. It is commonly called as flu, which is an airborne disease. So influenza, commonly known as flu. So we all are familiar with this. This virus belongs to the family Orthomyxoviridae. So myxoviruses. So originally it was myxovirus. Now it is called as orthomyxoviruses. So influenza viruses belong to the group orthomyxoviruses. They have RNA as a genetic material in which this RNA genetic material is enveloped. So it belongs to the family Orthomyxoviridae. Influenza virus belongs to the family Orthomyxoviridae and contains single genus, that is influenza virus. And there are three types of this influenza viruses or subtypes called as A, B and C. Now, these are classified under respiratory viruses. Influenza viruses are called as respiratory viruses. Why? Because they infect the respiratory system. So, what exactly does this influenza causes? So, influenza viruses. So, this is the name of the virus. The name of the virus is influenza virus, which belong to orthomyxoviruses. And the name of the infection is influenza or simply called as flu. So this influenza virus is an acute infection disease of the respiratory tract. What do you mean by acute infection? So acute is something which is sudden onset, right? So this is the meaning of acute infection, and it is a respiratory tract infection. That means, and it is also an airborne infection. The meaning of airborne infection is it is transmitted through inhalation of air droplets. So if an infected person, any person infected with the flu or influenza, so if that person sneezes or coughs and then releases the droplet nuclei, and that droplet nuclei consists of this viruses, and when an active person or a healthy person is going to inhale these droplets or droplet nuclei containing the influenza virus, this is the transmission mode of transmission, right? So yes, and it occurs, yes, this influenza virus, it occurs in sporadic forms or epidemic or pandemic forms. Sound. So this organism has caused sporadic, that means, so it is going to occur occasionally or at times or very rare or irregular instances, and also it also caused epidemics. So epidemic means an outbreak of any infection or disease which is going to spread very quickly and going to affect many people at the same time. And also pandemic forms. So we all are very much familiar with pandemic. One of the best examples of the pandemic is COVID or coronavirus. Yes, this is going to spread widespread infection of the disease over a whole country or throughout the world at a particular time. This is called as pandemic. So this influenza virus, it has caused sporadic, that means it occurred occasionally or irregularly at times, and it caused epidemics, that it is going to spread very quickly and going to affect many individuals at the same time, and also causing pandemics, that is throughout the world. So one of the important or unique feature of influenza virus, that it is ability to undergo antigenic variation. So we all know that infectious organisms agents consist of antigens, right? So this antigenic variation, so what do you mean by antigenic variation? So it is nothing but it is a change in the antigenic structure. change in the antigenic structure is called as antigenic variation. So you can see the definition here. So antigenic variation or alteration, it refers to the mechanism by which an infectious agent, it may be bacteria, protozoa, virus, which is going to alter the proteins or the carbohydrate on the surface and thus avoids a immune response of the host. So we all know that most of these antigens are either they might be proteinaceous or carbohydrate in nature. So the structure is going to change. So this structure, there is a change in this structure, and overall finally the antigenic structure is going to vary or alter. So that is how new variants comes into existence. So if you see the two types of glycoproteins or antigens which are present on the surface of the influenza viruses, hemagglutinin and neuraminidase. So if you see different types of influenza viruses, so they comprise of 4 different species. So they are influenza A virus, IAV, influenza B virus, IBV, influenza C virus, ICV, and influenza D virus, that is IDV. So A stands for alpha influenza virus, B is for beta influenza, C for gamma, and D for delta. So these are the 4 species of influenza viruses. Now this is the structure of influenza virus. So have a look at each part of this virus. So if you see here, so these are called as spikes, hemagglutinin, so or simply called as HA, and these are the antigens which are present on the surface. You can see that is hemagglutinin. So this is a lipid bilayer. This structure is a lipid bilayer, and we also have neuraminidase. So this light orange color is a neuraminidase, and we have matrix protein throughout. You can see here this round blue color is a matrix protein, and we have M2, that is ion channels. So these are ion channels. So here, so these are channels of ion, and then we also have nuclear export protein. So this one is a nuclear export protein, right? So this is a simple structure, and the one important thing is it consists of RNA as a genetic material, which is a negative sensed RNA genetic material. So you can see here this black color one which is there, so that is the RNA. So negative negative sense RNA strand, which is attached to NP or nucleocapsid protein. So this green color circle chains are called as nucleocapsid proteins, right? So this is inside this, so they are in the form of segments. So the RNA is in the form of segments, so that is why you can see like pieces or like segments, this RNA is present inside. So these are the spikes, hemagglutinin spikes and neuraminidase, which are responsible for genetic variation. So this is a 3D image of influenza virus. So you can see here the spikes, that is a hemagglutinin, and we have the ion channel, lipid bilayer envelope, neuraminidase. So this is a neuraminidase. So this is a 3D image of influenza virus. So coming to the morphology of influenza virus, so they are spherical in shape with a diameter of 80 to 120 mm, but also they show pleomorphism. So what do you mean by pleomorphism? So they keep changing the form, they keep changing their form. So that is the meaning of pleomorphism. So mostly they are spherical, but they may also be occurring in other forms, like sometimes cylindrical or circular or something like that, right? So you can, if you see here, yes, so coming to the symmetry and all, right? So we have seen in the previous image that the nucleocapsid is surrounded by an envelope. So it has an envelope, and the virus core consists of ribonucleoprotein, which is helical in symmetry, And yes, the RNA genome is of single stranded and is segmented and exists at 8 pieces. It might be 8 or 7 or 6 segments. Generally, the RNA or the genome is going to exist as segments, and the number may range from 6 to 8. Right. So it also has got an inner membrane protein, right, and outer lipid layer. The membrane protein also called as matrix or M protein. M stands for matrix protein. Yes, so the two types of peplomers, that is nothing but the antigens or the spikes are nothing but hemagglutinin spikes, which are triangular in cross section, and the mushroom shaped is a neuraminidase. Yes, as we have already discussed, the DNA or the genome is single stranded RNA which is segmented. Yes, as the different types of influenza exist, type A and B influenza virus consists of 8 pieces of segments of RNA, while type C consists of 7 segments. Yes, the surface glycoproteins are nothing but hemagglutinin and neuraminidase, which are important antigens which are going to determine the antigenic variation of the influenza viruses and the host immunity. Okay, so let us discuss about hemagglutinin. So the hemagglutinin, it derives its name from its ability to agglutinate the erythrocytes, right? So even this name, myxoviruses, so they belong this influenza viruses, they come under myxoviruses or orthomyxoviruses. The name myxoviruses has come from the ability of these viruses to adsorb onto mucoprotein receptors which are present on the erythrocytes. So on the RBC cells or the erythrocytes, certain receptors called as mucoproteins are present, and these viruses, they adsorb, they get attached onto that receptors which are present on the host RBC cells, hence causing hemagglutination. So that's why the name is referred to the affinity of these viruses to mucins. So mucin is nothing but the mycoprotein which is present on the RBC cells and hence leading to hemagglutination. So this hemagglutinin, which is a glycoprotein composed of two polypeptides as HA1 and HA2. Yes, so these are responsible for adsorption and agglutination. And these two HA1 and HA2 are joined by disulfide bond. So this triangular shaped hemagglutinin, so the shape of this hemagglutinin is in triangular shape, which is inserted into the virus membrane by a tail end and is hydrophilic in nature. So we also have some important points here. Okay, so I'm going to skip this, girls. Yes. Now let us discuss about neuraminidase. So this is also a glycoprotein receptor, and it is going to determine the subtype of the influenza virus isolates. So we have subtypes of influenza viruses. So this glycoprotein, that is neuraminidase, is important in determining its subtype. So based on this neuraminidase, the subtype is going to be determined. Subtype of what? That is the influenza, the type of the virus. So the shape of this is mushroom shaped spike kind of thing, this neuraminidase, and it's a tetramer composed of monomers. So the main function of this neuraminidase is going to be played at the end of the viral replication cycle. So you know that viruses, they are going to infect the host cell, and inside the host they are going to utilize the machinery of the host, and then that is how they are going to replicate and form progeny cells, and finally lysis of the cell and release of the progeny cells. So at this end, its main part is being played, that is in the viral replication cycle. So this neuraminidase is a enzyme. What kind of enzyme? This is very important point. It is a sialidase enzyme which is going to remove the sialic acid from the glycol conjugates. So we'll discuss this in detail in the next coming slides. So next coming to antigenic variations. So as already discussed, this influenza virus shows high antigenic variations. That means it is able to frequently change, and as a result, every time a change in the antigenic variation is nothing but every time a new strain is going to appear. So, so this is an important characteristic that it is going to regularly change the surface antigens, and we have discussed that the two surface antigens which are responsible for this antigenic variations are hemagglutinin and neuraminidase, which are present on the surface. Yes, now there are two types of antigenic variations: antigenic shift and antigenic drift. So let us discuss in detail about what this antigenic shift and antigenic drift are. Antigenic shift. So shift and drift. So shift is something which is related to a drastic or discontinuous change in the antigenic structure. As a result, it is going to form, a new strains are formed, a novel or a new one which was not there earlier. So you know the meaning of novel. So like for example, in the previous 2020 coronavirus, we have seen that it is a novel coronavirus. Even the coronavirus, the pandemic which resulted, it was not a new virus, but coronavirus exists previously, but the strain which caused pandemic was a completely novel one or a new one. So this is the meaning of novel, which was not present earlier, something which is very new. So antigenic shift is all about the major and drastic discontinuous change in the antigenic structure, which is going to result in novel strain, and it is antigenically unrelated to the previous strains. So there is no relationship between the presently formed and to the previous one. So it is completely different or a new strain. And what is the reason for this antigenic shift? This is mainly due to the mutations. So the major antigenic change which is going to occur in hemagglutinin and neuraminidase antigens is by the change in the genes or replacement of the genes which are responsible for coding of the hemagglutinin antigen. So it is all about the genes, the rearrangement, or so instead of one particular amino acid, it is going to completely code for a different amino acid. So there is a change in the sequence because of the exchange of the RNA segment. So this is the reason behind this antigenic shift. So you can also say that it is because of mutations. So mutations is nothing but change in the gene sequence. So instead of synthesizing one particular amino acid, it is going to, and the genes or the codes are going to change, and as a result finally the amino acid sequence is going to change, and that is how there is a change in the new strains are formed. Yes. Now the antigenic shift occurs when two different strains of influenza virus is going to infect a single host cell. That means two different strains. Say for example, what is the meaning of antigenic drift? So this is a gradual sequential change in the antigenic structure. So whereas we have seen the previous one, the antigenic shift is a drastic change, but this is a gradual sequential change in the structure of the antigen. So this drift, antigenic drift, it refers to the minor changes either in the hemagglutinin or in the neuraminidase or both together. So there is only a minor antigenic change which is going to take place in the hemagglutinin. There is no much variation. There is only a slight or a minor antigenic change. That is the meaning of antigenic drift. So because of this, so this is a very important feature of this influenza virus. That is, there is continuous change in the antigens of this influenza virus. Hence, every time a new strain is going to occur. Now coming to the mode of transmission. Yes, how is this influenza virus going to transmit? So the virus infection is going to transmit through contaminated air. So this is a airborne disease, airborne. So the route of infection is air, as through contaminated hands as well, and it spreads from person to person via respiratory secretions as well during the act of sneezing, coughing, talking and singing. But it can also be transmitted by contact with contaminated hands and surfaces, because this is related to respiratory tract infection. And in influenza virus there is coughing and sneezing, running nose and all. And when the virus is going to be present, so when the hands and all we are going to touch the face or nose region, and again those we are putting it on other surfaces, so that is how the transmission also occurs. So this is a mode of transmission. So this influenza is a airborne disease. Coming to the very important part, pathogenesis. So how is this pathogenesis occurring in this influenza virus? So the virus is going to enter inside the body through the upper respiratory tract. So it is in the upper respiratory tract, it is going to enter inside the body. So the route of entry for this influenza virus is upper respiratory tract, where it is going to attach to the mucosal epithelial cells. So in the mucous membrane, in the epithelial cells of the upper respiratory tract, it is going to attach to the epithelial cells. But as a result, this influenza virus also has got some defense mechanisms to protect itself. Okay, right? So IgA antibody, it is going to, which is present in the mucous lining of the upper respiratory tract. So this virus is going to neutralize this antibody which is present in the linings of the mucus of the upper respiratory tract. So that is a defense mechanism which is played by this influenza virus. Now as soon as it enters the mucosal epithelial cells, it is going to attach to the receptors of sialic receptors. The name of the receptors is sialic receptors, which are present on epithelial cells. With which spike, as we have seen that two important spikes or antigens, that is hemagglutinin and neuraminidase. But hemagglutinin spike is the one which is going to help in the attachment or adsorption to the sialic receptors on the epithelial cells on the viral envelope. So this spike is present on the viral envelope, as we have discussed and seen in the image of the morphology of the virus. Now, what is the function of this neuraminidase? So hemagglutinin spikes are going to help in the attachment to the sialic receptors on the epithelial cells of the upper respiratory tract, whereas neuraminidase is going to lower the viscosity of the mucus film. So we all know that the respiratory tract is lined with mucous membrane. So the viscosity of the mucous membrane is lowered so that it will be easy for the virus to spread or enter inside the cell. So that is the function of this hemagglutinin and neuraminidase antigens. The next step after attachment on the epithelial cell, the virus enters into the cell and it will start the replication cycle, or the life cycle of the influenza virus starts. Because viruses are intracellular parasites, they require a living host in order that they use the host machinery and replicate and produce progeny cells, and finally lysing the host cell and release of the progeny cells. So this is the process, general process of all the viruses. So as it is going to enter the epithelial cells, inside the epithelial cells, once it is entering inside the cell, it is going to start the replication process. So as it is in the mucous cells, in the epithelial mucal cells, destruction of the cell is going to occur and degradation of the mucous membrane. destruction of the cells, cellular destruction and degradation of the superficial mucosal layer occurs. Now, as a result of this, if you see, the symptoms include non-reproductive cough, sore throat, nasal discharge are generally exhibited by the person. Now, after this, the virus is going to continuously replicate in the epithelial layer of the respiratory tract, and as a result epithelial layer is completely removed because of the destruction of the cells. The respiratory tract is epithelial layer of the respiratory tract is completely removed. Now, as a result, we have also studied about the normal flora. This opportunistic normal flora are taking an advantage now, and they are going to enter or penetrate through these damaged epithelial cells and cause bacterial infections. So the important bacteria like Streptococcus, Staphylococcus and Haemophilus influenza, which are generally present at this region, are entering inside this destructed epithelial cell, and they are going to cause infection in the lungs. So here in the lungs, these bacteria are going to multiply and they cause secondary bacterial infection, because this infection is caused by the opportunistic normal flora which are present at that region. So what are the three important bacteria here? Streptococcus, Staphylococcus and Haemophilus influenza, and then they cause secondary bacterial pneumonia is caused here. Okay, so those who are immunologically compromised or other patients, I mean like lung or heart patients, so the infection may be severe, and in such cases alveoli also may be involved, and it may be severe. The pneumonia may be severe and completely loss of blood also may occur, that is lung hemorrhage or loss of blood also may occur. So here influenza is going to remain in the epithelial of the upper respiratory tract, and it is not going to penetrate into the inner tissues. That means it is restricted to upper respiratory tract epithelium only, and the virus is generally not found in the blood of the infected individuals, but it is going to remain in the epithelial cells of the upper respiratory tract. Yeah. Here is a diagrammatic representation step by step, which is showing the infection of the host cell with the what do you say, influenza virus. So you can see here this is a virion. So it is going to attach to the cell, and then here, so once it is entering inside, so inside the cell this is the endosome, and then transcription is going to occur. So using the machinery of the host cell, the replication of the RNA is going to occur. Replication of the RNA, then followed by translation. So that is how the bacteria is going to, the virus is going to synthesize the progeny particles, and finally this is how the virion particle is synthesized and released out of the cell. So this is a diagrammatic representation depicting host cell invasion. What is invading here? The virion particle. Influenza virus is entering inside the host cell. How it is going to replicate, synthesizing the progeny particles, and finally after the synthesis of the virus, they are going to be released out. Once they come out, they will go and start infecting the new cell, the fresh cell or the new cell. So that is about the image representing cell invasion, host cell invasion and replication by the influenza virus. Now, so the organism, that is influenza virus, is going to reside in the upper respiratory epithelial cells but not found in the blood. Now, what are the clinical symptoms or manifestations of this influenza? So, generally, we are familiar with these flu-like symptoms. So, generally, whenever you say flu-like symptoms, with what all will come? So, the classic influenza symptoms will include chills, headache, dry cough, high fever, muscular aches, malaise, and anorexia. These are the common symptoms which are found in the influenza. Sneezing is common, rhinorrhea, and nasal obstruction. So, just generally, this is observed in flu, influenza. But some patients may also have photophobia, nausea, vomiting, diarrhea, and all. So, fever is going to remain for 3 to 5 days, and the respiratory symptoms are going to last for 3 to 4 days, and the cough and weakness may remain for 2 to 4 weeks. Now, coming to the clinical symptoms of influenza, children are similar to those adults, although children may have fever and higher instance of gastrointestinal manifestations, that is vomiting. Vomiting is also seen in case of children. Okay. Coming to pneumonia. So as we have seen that because of the destruction of the epithelial cells, upper respiratory tract epithelial cells, the normal flora are going to become opportunistic and they are going to cause bacterial pneumonia. So, as a result of this influenza, serious complications of the influenza usually occur in the elderly and immunocompromised individual, especially those with underlying chronic disease. So, those who are immunologically compromised and those who are elderly, in such people, this there are certain serious complications which are caused by this influenza virus, and not only that, the pregnancy women are at a higher risk for lethal pulmonary complications, generally in some epidemics. So this is the image which is representing the symptoms of influenza, that is headache, fever, muscular pain or extreme tiredness, joint aches, vomiting, coughing, sore throat, aches, runny nose, and stuffy nose. So coming to the lab diagnosis. So generally, because this is a respiratory tract infection, the samples which are collected or used for diagnosis of influenza virus are nasal or throat washing or throat swab. Gargles also can be collected, which should be collected within 3 days after the infection. So serum sample also can be used in case if antibodies which are present in the serum, antibodies against this influenza virus, if we are detecting, serum sample or blood sample can be collected. So coming to the diagnosis part, so antigens which are present, yes, direct antigen detection can be done through this nasal washing or throat swab. So these antigens are nothing but the components, that is the parts or the components of the viruses during the assembly replication. So they might be left out during the replication process, some of the parts, the antigens. So that can be found. So these are nothing but antigens, that are the components of the virus which are left during the assembly of the virus particles. So in the nasal swab, throat swab, so where the virus was dividing, so from there these antigens can be isolated. So other rapid diagnosis of this influenza include ELISA or FAT, that is fluorescence antibody test, RIA, radio immunoassay using specific monoclonal antibodies can also be done. Next, coming to isolation identification. So the virus can be isolated from the patients, and then it can be cultivated in eggs like monkey cell culture or in the eggs, Um, it can be inoculated directly into the amniotic cavity. Also, it can be inoculated into amniotic fluid or allantoic fluids, and the cells after growth, particular growth, that is for 3 days at 35 degrees Celsius, they can be harvested and tested for hemagglutinin. Coming to the serodiagnosis, antibodies to several viral proteins like hemagglutinin, neuraminidase, nucleoprotein, and the matrix, lipid bilayer, and all. So, the antibodies to these antigens, which are produced during the infection with the influenza virus, will be present in the serum. So, that can be detected. How? Using serodiagnostic tests like hemagglutination inhibition assay, ELISA, fluorescence antibody tag, and that's how we're going to detect the antibodies which are specific to these antigens. So these are the antigens here. That is the hemagglutinin, neuraminidase, nucleoprotein, and matrix. Now molecular methods include RT-PCR. So RT-PCR is the genome of this virus is RNA. So that is nothing but rapid test for the detection of influenza RNA is the sample, that is we can go for RT-PCR.